Thanks, Rusty. Some of these introductions sound like your obituaries. But you might well hear about what people say while you are alive. What's the point when you're gone? I'd rather not be called father of gene therapy, but perhaps best is to describe as an early practitioner of technologies needed to advance gene therapies. So let me start out to give you a little bit of a general introduction what we do and how we got into it. So gene therapy, from my point of view, is an idea of introduction of genes in a cell, tissue, or an organ to correct a genetic defect or to cure a disease, ameliorate a disease, or slow down the progression of disease. That's how we started out thinking what gene therapy will be. Initially, gene therapy was really pretty much, we were considering having just genetic diseases because we thought the way to do that will be there's a defect and one can come back, that's fine, thanks. So for genetic diseases, it's easy to understand. You have a mutation, you put a gene which is corrected, and it can correct the defect. And we were quite excited, but really the excitement came that gene therapy actually offers an opportunity to influence the outcome of an acquired disease. For example, cancer, cardiovascular disease, neurological diseases. So in fact, I would bet there isn't an aspect of human health that cannot be influenced by introduction of genes. So the concept of gene therapy is remarkably simple. Introduce a gene, and its product should have alleviated the defect. But the problem is, how do you execute it? How do you introduce gene in a liver which has billions of cells? How do you introduce gene in a brain which has trillions of cells? How do you introduce gene in a hematopoietic stem cell which is 1 in probably 10 million cells? So it's not the concept, it's the execution of the concept that has delayed the idea of gene therapy. And in this case, most of people have spent a lot of time to figuring out what is it? How would you introduce the gene? Well, there are methods called physical and chemical methods, directly introduction of the gene, nanoparticles, liposomes. They're all great ways to introduce gene if you're doing experiment in a tissue culture dish. But the idea that you can actually have high efficiency to be able to introduce gene in a billion or a trillion cells, well, one day it will be possible, but we are not there. So most of us have concentrated on biological methods, which is mostly the viral vectors. Wide variety of different viruses. They could be integrating viruses, they could be non-integrating, they could be different types of vectors. And they all have, of course, issues with safety, toxicity, and of course, how big a gene can you put in? Because something like muscular dystrophy is almost 15 kilobases as compared to many other, which may be 1 or 2. So there are limitations for that. The basic idea for us, actually for gene therapy, really was using viruses. And the reason was the following. Viruses have only one thing in life to do. And what they want to do is to grow and to grow more. The viruses have two parts to it, a component which is needed for more to grow and a component which causes the disease. And the argument we made was very simple. Let's get rid of the component that causes the disease, introduce a therapeutic gene, keep the part that is needed for virus to divide and propagate, and the virus actually doesn't really know that I don't have the disease-causing entity, reconstitute a virus which behaves just like the starting virus, except it has now the therapeutic entity to it. And viruses can be made into billion or trillion virus particles per milliliter. So you can actually imagine possibly putting 1 ml of the virus of a titer of 10 to the 12, 10 to the 13 has formally the capability of introducing gene in every liver cell. It doesn't happen, but that's at least a capability. So that's why we all concentrated on the idea of viruses, because the high titers, the large amounts you can make. Now, I got into it. I showed this slide the very first day, and the argument was, if you recall from my first talk, that these two sequences, we argued that this must be a sequence of the sarcoma virus, and this is a complementary sequence of Maloney, and that they two don't match each other. That's why you have this structure. So I argued at that time that could we remove this sequence here of the sarcoma virus and substitute it with a gene of interest, recreate a virus which now has the ability to behave like a virus, but instead of causing the disease, the sarcoma, it will actually introduce the gene of interest. So at this time, I was joined by Dusty Miller, who at that time had just come to us from Stanford, and he had been working on APRT and other genes. The very first experiment, as Rusty just mentioned to you, was done in collaboration with Ted Friedman, who had cloned the cDNA of a disease called Lesch-9 syndrome, where it's an HPRT, the cDNA. It's an X chromosome, but more importantly, it has a selectable marker, so you can give the drug in the cell, in the absence of which the cell will die. When you put it in, they will survive. Or you can put HPRT. I showed this slide for two reasons. One, it's almost now 1983. That was the first paper, actually, that anybody actually introduced a gene other than there was some work on SB40, but it's the first one to do that. And in fact, I was quite amused. For the first time, we never realized it was in the New York Times, which is something which never happened, used to happen, but it was quite exciting. And that also made me feel maybe it is an important experiment. I showed this to you because 3/4 of the paper was just to show how we made the vector. Now, it's not even in material, it's in supplemental materials. That's where we put it. But that's really was a big, and I was good at making vectors. I loved coming to the lab, putting my lab coat, getting the ice bucket, putting the molecules and putting them together. That was fun. And I'm hoping when I'm unable to do my own lab, maybe some assistant professor will hire me as a postdoc. I'll make vectors for them. So here we made a vector. First time taking a retrovirus, where at this end of this we put the HPRT gene. And shown here is that if in the case of the rat cell which we transfused, there was no HPRT. When we transfused them with the vector, we can actually make HPRT. And that was really exciting because this was the first time we could actually make a protein in a cell where we introduced it with a virus. And there are all kinds of nitty-gritties here, which is not important today. So what we had done is we had taken a mouse sarcoma virus, removed the sarcoma gene. It's replication incompetent, but in order for it to divide, we mixed it with mouse leukemia virus, which then made the virus. But we don't want to have the viruses which can replicate. So at this time, actually, we were, once upon a time, we used to have many meetings together with people at Hutch. And Maxine Lineal had identified a packaging signal at the 5' end of an avian virus. And that was the signal needed for RNA to bind to get into a virus to be able to get into it and replicate. If you remove that signal, the virus will make all the proteins, will constitute, will make a virus, it will infect, but it cannot replicate anymore because the viral RNA cannot be packaged. So Richard Mulligan and David Baltimore and Mann had done the same experiment earlier than us in the case of the mouse leukemia virus, but very shortly after that, we did the same thing with making an amphotropic virus, which Dusty Miller actually used for the first clinical trials in ADA. At this time, we were joined by a couple of other interesting folks. Here is Dinko Valeria. Here is Lee from Australia. And here is Jackie Johnson. She joined us from Stanford. And also Dan St. Louis from Canada. And we finally began to form, this is in the mid-'80s, a group of people to start thinking about using different kind of ways to introduce gene using gene therapy. And we got into the first thing was a factor 9, a clotting pathway, where the factor 9 deficiency does not allow the blood to clot, and the children will die of bleeding. In fact, this is the one which was in the royal family in England, was the deficiency of factor 9 called Christmas disease. Factor 8 is more common. This is like 1 in 30,000, and it's X-linked, it's male, and this is actually more common, 1 in 10,000 is the factor 8. So you might ask, how did we get interested in hemophilia? Well, it wasn't that I knew anything about hemophilia. It wasn't that it was particularly interesting, but remember, we are talking now of the mid-'80s, when it was no DNA ligases, there were not many restriction enzymes, there was no PCR. I met George Brownlee in Cambridge, and he said he had been working on factor 9, and the only thing that struck me that the cDNA he had a BAM H1 sites at either end. That's the sole reason of working for hemophilia, because we could cut our plasmid with BAM-H1 and put it in and had nothing to worry about it. Now, this sounds stupid, but this is all, there were 6 restriction enzymes, or 10 maybe, so we needed to do these kind of experiments. So we got into it, which turned out to be a good thing because it's a secreted protein and a lot was known about it. So we began to make the first set of experiments where we'll put the factor 9 and put it into the fibroblasts and put collagen in it and implant into the mouse and ask the question now, will this mouse continuously make factor 9? And here it is, our enthusiasm is rising every day, peaks up, a New York Times article we were hoping, and it came dwindling down. And it was a surprise to us because in a tissue culture we could maintain the production of factor 9 for months, if not years, but we went into the animals, it completely shut off. And we realized later on that you need a specific promoter when there was a heavy amount of methylation of these cells because we really didn't know at that time. But this led us to start thinking about other kinds of vectors because it was clear that retroviruses only infect dividing cells. And we could only take fibroblasts out and grow them and infect them. We couldn't really do them liver cells, hepatocyte, brain, and other in vivo because they will not infect a non-dividing cell. So at this time, the adenoviruses became popular, and in fact, they could be raised into high titers. So we start asking the question, what if we put factor 9 in adenoviral vector, which was deleted so it cannot replicate? And this was great here because you can see the amounts we are making is almost 1 to 10 micrograms. Average human makes about a microgram to 5 at best. In the best case circumstances, 0.1 is sufficient to alleviate the problem between 0.1 to 0.5, and we could make 10 microgram of it, and eventually it went on for about a year's period of time. This is really exciting, except the astute among you might notice the experiment was done in a nude mouse, which means there was no immune system in this particular case, the mouse. When we did the same experiment in a wild-type mouse, because after all, humans are not immune deficient, once again, high production, and very quickly our enthusiasm went down. And the reason for that is that adenoviruses are highly immunogenic. We didn't know those things at that time, and it became very clear then. This is one of our first immunology experiments where we took the virus, treated it with sorlin so that there's no replication of the virus anymore, but it could still infect, and it couldn't divide. No proteins were made, but in the incoming viral proteins made sufficient amount of CTLs, meaning the cells were being removed where the virus was being produced. So we really could not use adenoviruses for long-term production. Now, Cloda and others have made new kind of vectors with different serotype, which allows you to do some of these things, but clearly adenoviruses are wonderful as tools. They're very good for short-term, but they're not there for long-term production of the protein. So we began to move on to another vector at that time called AAV. AAV is a very small vector virus called adeno-associated. It has 2 proteins called the rep and the cap, which can be removed and substituted with a gene of interest, in this case factor 9, and the proteins can be supplied by adenoviruses, which can then be separated on a gradient. So these are the AAVs, by the way, lots of people are using in neurosciences these days, and at that time they had just started coming out around, I think it's 88 or so. So we made this vector, and at this time we began to think that we need to really have mouse model systems. So we made the first factor 9 knockout mouse, which Lily Wang made it, and these mice now have factor 9 knockout, so we could actually begin to introduce the virus and ask the question, do we actually cure the disease in these factor 9 knockout mice? And you could see here, if you take a wild type, and APTT just stands for a marker for showing that you can actually have the formation of the fibrins so that you can actually clot the blood. And you can see 6 out of 6 mice survive. So what we did, you cut the tail of the mouse a little bit, not too much, just a little bit, so that then you look at the bleeding and the amount of time it takes for the bleeding to stop. It's not a great assay. You had a little filter sitting on the bottom of it, and you see when it stops. So the wild type, of course, all of them survived. Hemophilic mice, the one that we have generated, none of them survived. But those where we put the virus now and then cut the tail, they all survived. So it was clear. Don't feel bad, just a little we cut. Not too much. Just a little. And we give them a little injection, so they're not so bad. Julie is feeling terrible. That's why she works on plants, I think. Yeah. So with this now, we said, okay, before we go to human, we could now go to a larger animal. And we went to dogs. I mean to say, we started working on to the dogs. And this is a Kegel. It's a cross between beagle and a terrier. It's a colony which Dr. Brinkhouse kept in University of North Carolina since 1947, the year of my birth. And these have only one mutation in them. They have about in their factor 9. And as a result, they can't clot their blood, so they have to be constantly given boost of the purified blood. And Yifeng Dai did the experiment. And so we went to University of North Carolina, injected them with this virus. And this was really quite exciting. The wild type had no bleeding incidence. C52, 55, these are the names of the dogs. It's an expensive experiment because dogs are hard to maintain for a long period of time. And this one had 2 episodes, none. D39, I'm told, is still alive. So all these years of the time, it hasn't bled for a long period of time. So clearly it worked. So we were really excited. We think this is the time now to start thinking of a human gene therapy. Now, we don't work in a hospital. We don't have patients. But other people, of course, are much more interested. And one of the group of people was Kathy High and Mark Hay. They made these vectors, introduced them into a human for the first time who are factor 9 deficient. And unfortunately, this is what happened. Factor 9 was made at large amounts, with some amounts, but then it went down. And in fact, there was a lot of liver toxicity, as you can see, by the high expression of these enzymes. So suddenly, it became an issue. What seemed to have worked in mouse, what worked in the case of the dogs, failed into the humans. And the reason it turned out is that 70% of the people in this audience has antibodies to AAV2, which is the vector they used. So they kept on using higher and higher amounts to get expression because the virus was being rejected by the antibodies. And as a result, there was toxicity. And more importantly, they make CTLs at some point because the virus wasn't exactly what they needed to have. It has improved enormously now. The trials have been done now. Now there are dozens of people who have been treated with this factor 9. There are human diseases which are treated with Hammer9 in the brain and for the eyes. So AAV vectors are wonderful in cells where the cells don't divide like neurons and so on. But that was the beginning of these vectors, which were started by Jude Smolsky at University of North Carolina, and we got some of those very early on in the labs. So at this point, in 1994, this is what Rusty was describing, we had retroviruses, no good, because they can't infect a non-dividing cells. We had adenoviruses, which have serious immunological issues. We have AAV, they had immunological problem, but more importantly, the only amount of information that they can put in is about 3.5 kb. And most of the genes we were working on was larger than that. So we decided at that time to draw on a piece of paper what would we need. In fact, Nature asked me to write a little article to see what would be an ideal vector. And we designed this vector. We think an ideal vector will be one where we will be able to make billions of virus particles. It's good to make 20, 30, that's okay, but that's not enough to do the job. We get a paper, but not a job. Transduced dividing and non-dividing cell, whether it's a cancer cell or whether it's a neuron, whether it's a hepatocyte or whether it's a hematopoietic stem, whether it's a prostate cell, we should be able to infect any number of cells. It should be convenient to produce, that is to say, it can be made in large amounts that we want and easily. Introduce the gene in the genome where we want. So when we put these vectors, particularly based upon retroviruses, they integrate all over the genome. And of course, you knew that there was already problems with certain children who are with ADA disease because the virus was then going at the wrong places. So we need to know where it went into the genome, and the genome is 3.5 billion nucleotides, so you need to know that. We would like to produce the gene product is controlled, like a rheostat. Turn it on, turn it off, when and how much. We would like to have large genetic material in it, 4 to 10 kb. We like to have a target, which is a desired cell type, so that if we want to put the vector, it should go in the liver or in the brain. So it'll be nice to have that. And finally, we wanted to have no unwanted or unpleasant immune responses. So we're asking it a lot. We're asking it to behave like a retrovirus to integrate. We are asking it to integrate at the right place. We are asking it to turn gene when we want, and we want it to behave like adeno can infect non-dividing cell and yet have no unpleasant consequences. Well, at this point, you can only ask for the following, what Paul Ehrlich said. You have to have skill, patience, money, and luck. Well, we thought we have some skill, none of this. We have a little of this, and we were hoping we'll have this. And it came, the luck came in an entirely unexpected quarter in the form of HIV. Now, you would think this guy is a nut, because why would you think HIV is a lucky case here? Because that is one of the most pathological viruses we have around. But it came very clear to us that HIV is a virus which is much like a retrovirus. It has the ability to integrate into a non-dividing cell, because we realized neurons were infected with it, macrophages were infected with, non-dividing T cells were infected with. So therefore, we knew it has the ability to do that, which was missing in a retrovirus. So we started mixing and matching, and nothing happened. And there's a reason for that. I can tell you more in detail. At this point, Luigi Naldini came and joined the lab. And we began to ask the question of, so a group of people were formed here. Luigi Naldini came from Italy as a postdoc. And here is Didier Trono, who was then an assistant professor at the Salk. But he had a P3 facility where we could actually have HIV growing. Because that was a very big deal there. Ulrika Blumer was a postdoc in Rusty's lab, and of course, here's Rusty. I showed this slide to the Congress when I first talked about gene therapy to them, to show them that how American science is so much dependent on immigrants. Here is Italian, German, Swiss. I was born in Bronx, not quite really. And the only American hostage we have is Rusty Gage here. It is true. It is true. I'm not joking, because it is a fact that we have a lot of immigrants, at least, who do science. So this is a slide actually I showed, and Senator Ted Kennedy was shaking his head like this. So here is the vector that we finally ended up with. So what we needed to do was to get rid of the genes that were all involved in pathogenesis of the disease one way or the other. There are about 9 of them. And all we really needed was to remove them, provide these genes outside separately, and introduce a transgene over here. And we made such a vector, but we got only 20, 30 such particles. And I told Luigi, was very depressed. I said, Luigi, if we have 20, that just means we have to make it now 2,000 or 20,000. And Ted Friedman's lab had just shown that if you used a glycoprotein from vesiclostomatitis virus VSVG, you can get high titers because you can spin the virus, because the envelope protein doesn't fall apart. So we got that, and that certainly allowed us to make titers of 10 to the 6 to 10 to the 9 when we start concentrating. That is 1 billion virus particles per mil. And the vector finally that we had 9.9% of the genome left. 91% of the genome was essentially gone. So all those people who were scared of HIV suddenly begin to realize that the vector doesn't have anything to do with HIV, because we got rid of everything, gutted it completely, and introduced the gene of interest and provided these proteins separately. None of them was ever needed for replication of the virus. It's actually quite amazing to this date. Pretty much everyone who uses lentiviral vector uses exactly this very vector, pretty much. They have their favorite restriction enzymes and so on. And in fact, I'll come at the end, the clinical trials are using exactly the same vector. So we were very lucky that we could construct a vector which has at least the ability here to be made in high titers. The question now was, would it infect non-dividing cells? How efficient it will be, and can we use that? And here, the first thing we did was to show, again, because Rusty and I were next to each other, the easiest thing to was, is if we could show that the neurons, which are truly post-mitotic cells, can be transduced with this vector and produce the protein, then we are home free to say that we now have a vector which has the ability to introduce into non-dividing cells. So we injected it in the brain, and you can see that when we have the HIV vector, here is the GFP protein, and when we use our traditional mouse leukemia viral vector, we had no expression. This is actually, always when I see it, I have a pang of sadness, because 15 years of my life was devoted to MLV vector, and all it now serves is as a control. That's life. We then moved on to do a number of different things to ask. So this is with Takahashi, who was a postdoc with Rusty at that time, with the hero in my lab, to show that eye is a wonderful tissue, because if you can knock out eye, the mouse still survive because it's an external tissue. And these, we wanted to show that if you can introduce the gene, either in the photoreceptor cells or in the subretinal epithelium, will we have the production of the protein? And the answer is yes. If you put just a CMV promoter, which is sort of a macho promoter, doesn't care wherever it goes, expresses it, you see the expression retinal pigment epithelium. But if you use a rhodopsin promoter, which is strictly true for the photoreceptor cell, you see the expression in the photoreceptor cells. So with that, we took a disease called retinitis pigmentosa, which has about 40, 50 different mutations, but 7% of them have a mutation in the phosphodiesterase beta. So the argument was made that we make a lentiviral vector, put phosphodiesterase beta, take the mouse which has a retinitis pigmentosa, introduce in this, so here is the mouse, it has lost all its photoreceptor cells, introduce the gene, and ask the question, do we recover some of those photoreceptor cells? And shown here is that we have now many of the options. They did not actually start seeing. They were blind mice. They didn't actually become, they couldn't see, but at least it's a start. And now this is actually being used in a number of different patients in England and different parts of the world using different kinds of diseases in the eye. Muscle. Muscle is a tissue which is majority of our body, big parts of our body. So muscle is a very good tissue. If we could introduce genes and they could produce the protein, it will be a great factory. So shown here is a direct injection in the myofibers, and you have positive for the foreign gene. Liver. A large number of metabolic diseases of human are in the liver. So the question is, we would go directly into the hepatic artery. By this vector, do we have the production of the foreign protein? Shown here is an experiment done by Alex, directly introducing the gene into the liver, and then we can ask the question whether these liver cells are able to produce the protein, and we had a CCD camera to look at it. And shown here is again that the liver is, within 48 hours you have the expression. You have some in the spleen and other areas, but most of it's in the liver. Of course, the champion is really human hematopoietic stem cells. Think about it. Very difficult to transplant any other human tissue. The liver, the lung, the brain of course, not possible. The one that is constantly able to do is that we make blood every day. You and I make 10 to 20 billion cells a day. The reason is because there is a master hematopoietic stem cell which can continuously go on to provide the other cell types. And if we could find that stem cell and introduce the gene there, wouldn't it be great, because that can continuously produce the foreign protein forever, as long as we live, as long as we make blood. The problem, of course, is such a cell is very rare. There may be 1 to 10 of these in 1 million cells. If I have to take 1 ml of your bone marrow, there are 10 million cells. Maybe there are 10 to 100 of these. To find them is very difficult, right? You couldn't do an injection or something like that. And they go on to make all your different cell types, the myelin lineage, all your lymphoid lineage. So this is, I think, one of the major contributions that the lab made, was to start thinking, can we use lentiviral vectors to introduce genes into these hematopoietic stem cells? So the experiment was done. We took the cord blood cells from human, enriched them for stem cells, not quite fully, but reasonable enrichment, transduced them with the vectors, subliterated mouse, reconstituted, and asked the question, do they make now the foreign protein in their cells? And this is quite exciting. If we take our typical MLV vector, we see hardly any production. But if you now use our HIV vectors, you could see that in the bone marrow or the spleen or the peripheral blood lymphocyte, you make tremendous amount of the foreign protein. So that really sets the stage now to use hematopoietic stem cells for a wide variety of diseases, and that's really the most successful. There are over 700 clinical trials now, and I'll show you one of such example. This is not my work. I just want to show you, this is the work of a group from France. And I tell you this story because when I was the president of the American Society of Gene Therapy, 2 young women, 2 sisters, Rachel and Amber Salzman, came to me. And Amber said, I have a son who has this disease called ALD, which some of you might remember was part of the Lorenzo's disease, which was a movie at one time. And can I help them? Because it was shown previously that if you do hematopoietic transplants, that is to say, you take transplantation of their bone marrow from young children or a family member, put them into the other, the cells will transfer some of them to go into the brain, make the protein, and dissolve those plaques, that is to say, in this case, the lysosomes, storage disease, and the children behave perfectly fine. But she had no matching people in their family, so she wanted me to know, is there a way we could make them a lentiviral vector, which could then be used to transduce the blood, the hematopoietic stem cells, and then they could do that. Now, I had never seen the data, but the group in France had done that in the case of the mouse. So we actually met 2-3 days after the 9/11 in London to actually go through the data and agreed that maybe we can help them. So I was founder, like with Rusty, of a company called Cell Genesis. So we made a pitch to them that would they be willing to spend lots of money to make enough virus so that we can give the trial to these patients. So they were tried, given the first trial for those patients. And you can see here, these kids at diagnosis have a lot of problems in their brain, big myelin sheath, white matter over here. But once they were treated with the lentiviral vectors, and some of them are now pretty fine. They're in the schools, they're studying, they have no such disease left. So this is really very exciting, very first set of experiment. We didn't do it, but we were able to help them out in getting the virus being made. Luigi Naldini, the one you saw before, has gone back to Italy and is now really one of the world's leader in using hematopoietic stem cells for a wide variety of diseases using lentiviral vector. I just show you one quick one. 2 diseases here. One is called the ARSA, it's the MLD, and the other is another one, VASP here. These kids have a terrible, they can't stand up because they have this terrible big sulfites in their brain, and they eventually get rid of all their myelin sheaths. They have no balance, nothing left for them. So these kids have really hard life. But here, Luigi was able to introduce to 7 such kids, and you can see they're far improved at the age of 7, and they can walk, and they're making the protein for a very long period of time. In fact, there's no reduction in it at this moment. So this is very exciting, and every day you hear now of this kind of a thing. And for scientists like us, who started out as a curiosity that we can put GFP in a non-dividing cell, this is a long way, and I think this is the kind of a gratification we all hope we will have in our lifetime working in science. Of course, the exciting part in the case for us is that can you introduce these genes in the brain? The reason we did it, and this is why we got into glioblastoma, is because we had learned with Rusty and his colleagues how to introduce genes stereotactically in various parts of the brain. And together with Liza Maslia, we were interested in the very early experiments nearly 15 years ago on Alzheimer's disease, where you have amyloid plaques, neurofibrillary tangles, and neuroatrophy. So he had shown that there's an enzyme called neprilysin. If you could introduce that into the brain where there are plaques, you could dissolve these plaques with neprilysin. So we went into the brain, directly injected it into here with a stereotactic injection. This is just showing that you have expression of the gene, in this case happened to be GFP. But look at this now. If they have amyloid plaques, if we had lentineprilysin, the plaques are pretty much dissolved. Now, the next question was, how do you put such a protein which can cross the blood-brain barrier? So we then, work of Brian Spencer in the lab, begin to ask the question, how do you cross blood-brain barrier? Well, we knew there are many receptors which are bound to these endothelial cells, like LDL receptor, to which apolipoprotein binds, which can cross the blood-brain barrier and go into the brain. So we figured out that there was a small sequence of about 39 amino acids in ApoB, which was needed to be linked to a protein, and the cargo can then go across. And shown here is the way to do that, is that if we have now apolipoB protein, which we made a lentiviral vector, put it in the liver, the virus was secreted, crosses the blood-brain barriers. Here's the calbendin positive recombinant protein, LDL receptor, and here's the merge. So this is a very successful way of introducing a protein which can cross the blood-brain barrier. And we did this in a number of different ways. Of course, what we would like to do is to go back into the mouse, which has the amyloid plaques, because we can't just put a needle in every place. It would be nice if we could just do it systemically. And shown here is, again, is neprilysin with the plaques here, and here once we put our ApoB sequence to it, there are far fewer plaques in it. This also allowed us then to begin to start using these vectors to see if we can actually block the expression of certain enzymes. In the case of the Alzheimer's disease, you have the secretory pathway, the beta. First you have the beta secretase, then the gamma secretase, and that's how you end up making the amyloid plaques. So the question was, gamma is very hard to do because it's involved in many other things. Could we block the beta secretase? And in those days, shRNA is what was new, not like the CRISPR days. At that time, we have shRNA. So we made a lentiviral vector making shRNA to beta secretase and went into the mouse, which had plaques in it. Here is the APTT mouse with the transgenic plaques. If we put a control, there is loss of the neurons in the hippocampus, but when we put the siRNA to base, they came back. So again, another way to introduce genes to show that you can have an effect in the amyloid. Of course, this led us to do the famous experiment with the water maze. I always wanted to do it because everybody else showed them. So we said we will do this experiment because our question is that the mice, which are Alzheimer mice, which has if we put siRNA to the base, they should remove those plaques, they should dissolve those plaques, they should not allow the formation of those plaques, and they may have a better memory. So the way you do these experiments, most of you know it, there is a water maze here with the color, of milky color here, and there is a platform. You teach all mice at the same time and ask the question, is this better than the other? And the important part here is really the cues that you give them because they know. I think I can probably show it here. So you can see this mouse has the Alzheimer plaques. They were trained at the same time, but it sees the cues and goes around and around. It will take a much longer period of time to find it as compared. It is going to go on. I'm going to waste my time, but just take my word. On the other hand, when you look at the one which is now been given the lentiviral vector beta secretase, it goes on. The training was exactly the same, but it finds it very quickly with these cues around it, which actually reminds me to tell you about the rabbi and the priest when it comes down to the cues. Rabbi Moshe was a good friend with Father Graham. And one day he saw Father Graham, who looked very unhappy. He said, "What happened, Father?" He said, "You know, I was giving my sermons, and somebody stole my bike." "Oh, that's terrible," the rabbi said. "You know what? Next week when you give your sermons, have 10 commandments. When you reach the 7th commandment, say, 'Thou shalt not steal,' quickly look it up. Anyone who's squirming, that's your man." The priest said, "Good." A week later, they saw the priest. Rabbi sees him on his bike singing the hymns of the Lord. And he says, "But Rabbi, you found your bike. What happened?" He said, "Did you do what?" He said, "Exactly what you said. But when I read the 5th commandment, it said, 'Thou shalt not commit adultery.' I suddenly remembered where I left my bike." It's okay. This is a doable job. So this is just showing you again the same experiment, but more in numbers here. That took us to the next bunch of things to ask the question, what about cancer? We have large number of cancers where there's actually a translocation of a chromosome. So could you actually prevent the translocation of chromosome, go with an shRNA, prevent the formation of just that messenger RNA protein? Could you be able to affect that? So there are many such translocations. The one we worked on is CML, because we were working with Owen Whitty, who had these CML mice, where we took many of these siRNAs and miRNAs and showed that after injection, the mice are pretty clear of their leukemias. So that's another way to start thinking about in terms of how you can use these vectors. Of course, the most exciting lately, which you probably hardly can spend a day without having one newspaper or telling you, is this new idea of immunotherapy. Immunotherapy is fantastic. It is working beautifully on large number of patients, particularly where you have a checkpoint deficiency, they give you different drugs. But in the case of the other way to do them is that when a cancer cell has a very specific epitope on it, you can actually design a CAR-T, which is a chimeric antigen receptor. But the problem is you can design a receptor, but how do you put it back into the T cell? Because there are a large number of T cells. So here they use the lentiviral vectors, precisely using these in the lentiviral vectors, and then transducing the T cells of the patient and putting them back. So all the companies, the Kite, the Juno, and Novartis, and Bristol Myers, they're all using exactly the same vector that we designed many years ago. And this is really, again, another satisfactory thing to know that that vectors that you made can be used in the clinics. And it's actually quite remarkable how well these things work. This is a patient who has a tumor, I think it's acute ALL, acute lymphocytic leukemia. It's terrible. You can see the scan. They were treated with the CAR-T against this particular epitope, CD19 in this case, and within a few weeks it is pretty clean. Within a few weeks. That's, it's again not our work, but it's remarkable what one can do with this. And in fact, Novartis is now selling this as a drug for $475,000 a patient, but this is the first time they're actually guaranteeing it. They say, if in 3 weeks the tumors are not shrunk, we will pay your money back. This is a new way of start thinking of an expensive drug. I don't know if it's a good strategy or bad, but at least they're so certain that it will work, because it is quite expensive to make the vectors and transduce these cells. These vectors are used in the lab now in a wide variety of ways. Here is a vector which Odette in the lab made, where you can put 6 different proteins on one vector, which is pretty impressive, because if you are trying to put 2, 3, 4 in your transduction systems, you can use these vectors and make them in fairly large amounts. Now, I am running out of time here. It's already 16:55, and today there is food out there, so I'm going to skip certain things. I was going to tell you the work of Dimi in the lab, where we are trying to actually do more in the liver. So we are actually trying to use the human liver cells, put them into a mouse to reconstitute the whole liver of the mouse with a human, so that we can then eventually go on to do that in the case of human. So here is a mouse where we regenerated completely. This is a human liver. And in fact, you can see that hepatitis virus can very easily transduce them. And when you give the drug, they come down. You give the drug, they come down again. So we have now ways by which we can actually completely resynthesize or make a human hepatocytes into a mouse. And now the question is, can we do the same thing into the human? Well, just to very quickly, the work of Shavashni, what she has been doing is to taking now cells from a hemophilia patient. So she takes blood from a hemophilia patient, makes into iPS cells, takes those iPS cells, and in this case now she is more sophisticated. She uses CRISPR to correct the gene defect, which we know in each of those patients. She makes them into hepatocytes in vitro. She takes these in vitro hepatocytes, puts them back into the mouse, and reconstitutes and show that these mice now, which were factor IX deficient, are completely recovered. So she made all these mice which has all the factor IX deficiency and FAR deficiency, and shows that they have the ability to make the protein. So this is something we are now pursuing quite extensively. I'm just going to go to the last bench here. So this is another way. Sorry, Suvashni, I didn't mean to really shortchange you, but the idea, I did actually. Please read her paper. One came out and one hopefully will get accepted. So what she has been also using is RNA as a way of introducing these genes. So what are the challenges for gene therapy? I drew this slide, I think, almost 20 years ago, and it pretty much still remains the same. We still have a major challenge of delivery and efficiency. You know, we are putting genes in liver for 1% or 2% cell. We need to do much better if we want to make sufficient proteins. We still have a problem of long-term expression. We don't really know how long and how to regulate the expression. There are no regulatable system as yet FDA approved, so that still needs to be done. There are serious immune problems. All these vectors will always have some problem with the immune system. Think about it. You're bringing in billions and billions of virus particles into organisms. And normally, in the humans, when the virus comes in, it's only a few particles, and they go hundreds, and they go thousands, but then the immune system kicks off. But here we are bringing billions of particles to start with, and that causes a lot of problems which will have long-term consequences for the organisms. We still know nothing about, very little about the cell biology. people are doing things. For example, we made hepatocytes. How are we going to put it back in the human? Are we going to do hepatectomy? Well, that's hard to do in a hemophilia patient. Where are we going to put it? Are they going to go in the bloodstream? We know very little. The only thing that we truly know the best, to be honest, is the hematopoietic stem cells. There you take the blood stem cells, put them back, and they go home right back into the spleen, and so you can make them again. But we need to do a lot more work actually in the cell biology of the system. So where we are. I don't know if Mark Monton is here or not. Mark always tells me that you need 10,000 hours of success before you achieve hard work before you achieve success. Well, there have been plenty of that here. We started as early as 1982, proof of principle. The first gene therapy trials were done in 1990, didn't work out because the patients got leukemia in that case. Then we had this terrible incidence of Jesse Gelsinger, who got adenovirus at a very high titer, and as a result he had a toxic shock to it and died very bad death because of very high levels of toxicity due to adeno, because that went into the liver at very high titer, wasn't good for them. That really set back gene therapy a lot, because headline in New York Times was gene therapy doesn't work but it kills, and that really is not something you want to read in the subject you're working on. Hemophilia trials didn't really do very well for a while, but now the success came. Now the adverse event of skids are gone, and currently, as I said, there are over 700 clinical trials, and this is a very successful venture of curing people of diseases for which there really existed no cure. Many of the patients have absolutely nothing for them because these are point mutations, many leading to permanent debility to the life, or some for a short life of the patient. But giving these viruses, not mine, many people all over the world, is I think a fantastically successful venue, something we started as a pure curiosity. And as Rusty said, this is how old I will be when it will truly become a daily routine. Now, I have shown many pictures of many people to you over the period of time, but labs don't run by postdocs. They come, they go. Some stay, but most of them actually go away. You need some people to run the lab. And people like me who are traveling around, also you need somebody in the feet on the ground. I have been very lucky that I had only 2 secretaries in my last 43 years. One was for 70 years, and now I have Beth, who really runs the show, who knows exactly what goes on and what needs to be done. So thank you, Beth. I have Mark here, who has been with me for 25 years as a lab manager, and it's amazing how effective Mark is. Nothing phases him. He gets things done, and I'm very fortunate, and thank you. And also Nina, who has been for now almost 17 years with me, and just always smiling. It doesn't matter whether things work or not work, he's always smiling. That adds to the positive. So I'm really very lucky, not only that I have such wonderful people, but again, I would like to thank all the students, postdocs, associate, collaborator, colleagues, and there have been probably 130 to 140 people through the lab. So I'm very fortunate, and thank you for coming here for my 3rd lecture.